Good afternoon, everyone. Thank you to the organizers for giving me this opportunity to speak about my work on large-scale imputation of epigenomic datasets for systematic annotation of diverse human tissues. This is a highlight presentation of work published in Nature Biotechnology earlier this year, and it was a companion paper to the larger Roadmap Epigenomic Consortium effort. So to motivate this work, there's been growing interest in using epigenomic data to annotate the genome, in particular the vast non-coding parts of the genome. The reason for why there's a growing interest is that genome-wide association studies are continuing to implicate large numbers of disease-associated variation falling into these non-coding regions. And epigenomic data has been shown to be able, in case, to identify biologically relevant cell types and to identify putative regulatory elements that might be associated with some of the disease variants. However, there's some challenges that exist that we're trying to address here is that there's no experimental coverage for many cell type and epigenomic marks that might be relevant. And the coverage that we do have could be noisy due to variability in terms of the environment, the genetic or experimental factors. And this is particularly the case as we're moving more into primary cell types, where we have few, if any, replicates. And what you see here are examples of some of the epigenomic marks, histone modifications, the signal for that, and sometimes we represent these as chromatin states, which summarize multiple different marks into calls of different types of regulatory elements. So what you see here is a grid that we started with as part of the roadmap effort, where the rows correspond to different cell and tissue and they're grouped into larger tissue types. And each one of these we called a reference epigenome. 111 were generated by the Roadmap Epigenomics project, and 16 by the ENCA project that had been processed to be in the same manner as the roadmap data. And then the columns here correspond to different marks. Most of these are various histone modifications, as well as the histone variant, DNase hypersensitivity, DNA methylation, RNA-seq. And as you can see, the project took 5 core histone modifications and mapped them in every single reference epigenome. And then there's been other marks that have gotten, some have gotten fairly deep coverage, others in a relatively small number of cell types. And we were interested in the question, given we had a grid like this, could we go to a grid like this, where you see in red is it represents, we have an imputation, a computational prediction of what we think that genome-wide data set should look like, just leveraging that we have those marks in other cell types, and then other marks in the same cell type we're interested in. And we're also going to impute those data sets that we have observed data for also. So why might we be interested in doing this? For one, it's hard to get enough biologic sample to map every mark in every cell type, and also there would be the cost and time issue. And then another somewhat more subtle issue is that when we say we have some mark mapped in some cell type, that usually means we've had 1 or 2 experiments done on it, but there can be a lot of variability. And when you impute something, you're potentially combining information from dozens of highly relevant experiments. So there's a statistical robustness that you gain through the imputation that could potentially lead to higher quality data sets. And even if you didn't want to use the imputed data directly and you still generated the observed data, the imputed data gives you a prior on what you expect that data set to look like. So then it enables some unique applications, such as I'll discuss, quality control. You can flag data sets for which the observed data doesn't agree with imputed data. I won't have time to talk about this, but you can use it to also identify specific surprising locations, as well as also prioritize marks that could be able to impute other marks well for mapping. So how do we actually go about imputing these data sets? So I've developed this method we're calling ChromaImpute, and it takes a regression-based perspective to this problem. And the features in this regression can be divided into 2 high-level classes of features. The first class of features are other marks in the same cell type you're interested in predicting for. So how we present these features are we have the signal for each other mark we have in that same cell or tissue type at the target position in every 25 base pairs left and right up until 500 base pairs, and then 500 base pairs every 500 base pairs till 10,000 base pairs. And then what we also have that's quite informative in cases is we have information for that same mark and potentially many other relevant cell and tissue types. Because we have this large compendium now based on the efforts of these consortium activities. And in particular, we represent these features as the average of the mark signal in the KNearest cell or tissue types. And the question is, how do we decide what are the KNearest cell or tissue types? And it turns out what we do is for each mark that we do have mapped in this target cell type, we would define a different set of the KNearest ones, both separately based on a global distance measure and then also locally just at that specific, around that specific target position. So one of the challenges in this problem is that we don't have any training data for that target mark and that target cell type which we're trying to predict. So what we do is we train based on all the other samples for which we do have the target mark mapped and restricting to the common set of features that can be defined in each one of those cell types. And then we apply those predictors in the target cell type. So we use regression trees as our specific regression function, and then we're averaging their prediction. So we're taking effectively an ensemble of regression trees. And we make these predictions at a 25 base pair resolution, except for DNA methylation, we do at a 1 base pair resolution, but restricted to CG dinucleotides. So this is giving you a visualization of one region of, in blue is observed data, and then in red is our imputed data, not using those specific observed data tracks. It's shown for each mark in one of the various cell or tissue types. And this is at a 2 megabase resolution. We can zoom in at a 2 KB resolution, even go further down. This is a 10 KB region, and you can see we can capture very high resolution features such as this nucleosome dip. And this is another more systematic visualization. What I'm showing you here are each pair of heat maps corresponds to one of the major marks considered. The top heat map represents the observed data, the bottom one, the imputed data. Each row corresponds to a different reference epigenome. Each column is one of 2,000 randomly selected bins, and we've clustered them based on the observed data, and then we show the corresponding imputed data below it. So you can see we're able to capture even some very cell type restricted patterns, a yellow or higher level signal. And we've also done more quantitative comparisons. So this is showing you, for each mark we were considering, what's its genome-wide relation of the imputed data with the observed data. And we're comparing against two stringent baselines. One is if we just took the mark we were trying to predict and generated its average across all the experiments for which we have. And then the best case, if we just use the single best reference epigenome in place. So this would sort of correspond to how somebody currently might work, where if they were interested in some mark and some cell type and it wasn't available, they would make it their best guess on what the most relevant cell type is. And even in the best case, we're able to do better than that using the imputed data for all marks. But I was also arguing that the imputed data could have some properties that are better than robust, better than observed data. And this can give you some sort of intuition of this. So I'm showing you aggregation plots of H3K4MA3 around transcription start sites. So H3K4MA3 is a histone mark associated with promoter region. And you see in red is the imputed signal, and blue is the aggregation plot for the observed signal. And there's a much larger variance for the observed signal. And in some cases, what I'm highlighting here, E082 and 081, were actually two different fetal brain samples mapped by different production groups here. So this large variance can't just be explained at the level of the biology. The cell type is likely due to various other confounders, but they have a much more consistent profile with imputed data. And this is a similar type of plot for H3K36ME3, which is associated with gene bodies and transcription. The red, again, is the imputed data. The blue is observed data. And we can quantitatively ask how well can we recover expressed gene promoters based on H3K4ME3 for the imputed data and the observed data, and gene bodies for the imputed and the observed data based on here. This is based on H3K4ME3. I mean, K36ME3, and this is based on H3K4ME3. And I'm showing you this as a function of the number of cell types in which the gene is expressed. So one concern might be that we're going to miss very cell type specific behaviors and only capture some of the most broadly expressed genes. And it is true that the imputed data does improve in performance as the gene is expressed in more cell types, but it's competitive, in some cases better than the observed data by these metrics. I'm doing this in terms of area under the RSC curve up to 5% false positive rate compared to the observed data. And this is another view which might give you additional insight into some of the nicer properties of the imputed data. I'm showing you a correlation grid between all the experiments, and they've been ordered or grouped based on their sort of known high-level biological relevance, whether it's like a brain or a ES-type cell type. And I'm showing you here the heat map correlations for the major marks we considered. In the top is observed and imputed, and again observed and imputed. And you can see a much clearer block structure corresponding to the known biology of the cell types. And just in white, we can't even compute these correlations because we don't have the data. And we can quantify this in terms of the area under an ROC curve, where we ask, can we predict pairs of experiments that belong to the same group based on the correlation? And it's significantly better for some of these marks, more broader marks. So sometimes this observed data is not agreeing with the imputed data, and because we have some confidence in the imputed data because of this disagreement, we can propose that we could use this potentially as a quality control metric. So what I'm showing you here is a distribution H3K36 ME3's agreement with between observed and imputed data sets. So there's some outliers here which have very poor agreement. And if we look at the aggregation plot, for example, this E104 has what's considered a very unusual aggregation plot for H3K36 ME3. This is much more typical, where it goes up across the gene body. And if we use annotated genes, it was also ranked worse, and this is out of the 127 samples. All the major QC metrics the consortium had been using before had ranked this as a very high-quality data set, including one metric which put it as the #1 data set. And the problem with sort of the existing quality control metrics that had been used was they were largely measuring signal to noise, but wouldn't necessarily recognize antibody cross-reactivity or label swaps, for example. Going back to the original motivation of the genome-wide association studies, we compared the enrichment of GWAS hits curated in the catalog for specific studies with the signal of H3K27 signal enrichment, and did a Mann-Whitney test comparing those SNPs curated for a specific study to the signal distribution in the catalog in general. And we saw greater enrichment based on the imputed data compared to using the observed data directly, suggesting that we're able to filter a lot of the noise and keep some of the more biologically relevant signal. And this is showing you what these points correspond to, labeled sort of the most significant enrichment for each of the traits of what cell type is coming up. And a lot of these make a lot of biological sense. And we can also learn richer chromatic state models here. This is showing you, based on observed data, we were limited to learning chromatic state model across all the cell types with only 5 marks. But with imputed data, we no longer have this restriction, and we now had a chromatin state model which we learned across 12 marks that we were most confident in their imputation, providing a very detailed systematic annotation of the human genome across 127 cell types based on these chromatin states. And we did various annotation agreements to make the case that this has better agreement. And sort of very quickly, the last point is that we can start asking how well can we predict other marks using a subset of features. So this is showing you how well we can predict each mark if we only use the features from the same cell or tissue types. And there's certain marks like H3K36 ME3, K9 ME3, and K27 ME3, which are very hard to predict using only same cell type features. However, if we assume we have a compendium of data, and for example, if we only had H3K18 settle and K79 ME2, we can impute those almost as well as if we had all the data. And in some cases, for just these 2 marks, we can have imputation performance that exceeds some of the 6 marks that have been prioritized for deep mapping. So just to summarize, I presented to you about the Chrome Impute that allows imputing epigenomic datasets, and it can be used to provide a more robust version of experimentally mapped datasets well. And there's a resource now representing more than 4,000 datasets and 500 billion predictions. And I want to acknowledge Manolis Kelth's work started while I was a postdoc in his lab, and I continued to work on it after moving to UCLA. And this was used data from both the roadmap and ENVOD consortium, and these people did a lot of the primary data processing. And here are the URLs of relevant. Thank you.
Questions for Jason? Yes.
Thanks for this nice work. I have one question regarding the prediction. So you mentioned in many cases, right, you are able to make good predictions, and you also show there's some variation between when you can predict, when you cannot predict, which is exactly what we expect. So my question is, does your approach basically tell you when it doesn't know about a prediction? I mean, do you have good confidence estimates that you can say, I'm only predicting this well, but these are my error bars, and if I go to the wrong cell type or the wrong context, these error bars are large, and they really tell you, okay, this approach is not able to confident predictions in a particular context, which would be probably a very useful thing.
Right. So the sort of the degree of sort of the height of the signal of the prediction is both a reflection of the confidence of the prediction and sort of how high it thinks is. So it's not directly giving error bars. When I was saying we have sort of the 12 marks we were most confident in, that was sort of because we have those marks mapped in a lot of cell types. Some of those other marks have only been mapped a couple times, so we're sort of less confident that we can learn effective things. But yeah, sort of a more statistically rigorous confidence prediction would still be an open problem here.
Thank you.
We have a question on the right here.
Hey there, great talk. What's roughly the number of files you need to impute all the rest in the encode data?
So if you're assuming you already have an informative compendium of data, then just having around like 2 marks could allow you to impute well into new cell types. But if you don't have any coverage of a cell type that's relevant, then you're going to need to do much more, 6, 7, 8 potentially, to if you didn't want to trust your compendium of relevant information.
7, 8 marks, and then all the rest is completely redundant with these 7, 8 marks.
I mean, it's all relative. I mean, there's still some information you're going to gain, but it'll be sort of diminishing returns.
Thanks.
The last question here on the left.
Great talk. Thank you. So you said you are averaging the predictors in computing your final prediction. It seems like if there are different tissues and some tissues are more similar, it's more like a multitask setting where you would want to upweight some predictors from tissues which are more similar. Isn't that the case?
So we're updating more similar tissues through the KNearest neighbor features. So that is how sort of the more similar tissues are getting upweighted. Sort of the assumption is that the correlation structure is relatively conserved across cell types, but sort of the position of the marks moves. So that's why we're not per se like emphasizing more similar tissues in how much we weight their predictions. But there potentially could be more work done.
So we have a few more questions. Please track down Jason in the break, and let's thank him again.